Hello students, in the last video we have seen the classification of animals. So we have understood the basis of classification, then classification also we have seen. Now let's just start with all the phylums. So the very first phylum in the animal kingdom is the phylum Porifera. So let's just start with the phylum Porifera. So phylum Porifera is the first most primitive group of animal, and this phylum Porifera, it will include around 5,000 species. So this 5,000 species are here basically in water. They are all aquatic. So first of all, just see what is that Porifera word means. Porifera. First of all, this means pore and fera, this means bear. So these organisms, they are pore bearing organisms, pore bearing animals. So around 5,000 species are here. The organism is a hollow structure, sponges. You can see the whole body will have some pores. Okay, so that is the phylum Porifera. So let's just start with the basis of classification. So first of all, those bases of classification we'll see in the general characters. So what are all general characters? Let's just see. First of all, their habit and habitat. So first of all, see habitat. What is the habitat of the phylum Porifera? See, when evolution took place, actually all the animals start to evolve. Not only animals, even the living organisms, they evolve from the seawater. So in the seawater itself, all the nutrients, all the necessary things, environment were present. So that's why life started actually from the seawater, and gradually it became terrestrial. So what will happen in the phylum Porifera? These are also aquatic. You can see also whenever we'll see it or we'll start studying all these phylums, then we will get to know that primitive organism actually are the aquatic organism, and gradually it become terrestrial. So this is the most primitive group of animals. So that's why they are aquatic organism. Maximum they are marine. Mostly they are marine, but there is an exception, and from that exception question comes. What is that? There is a freshwater species are also found. So what is that freshwater Porifera? That is Spongilla. Remember, Spongilla is imported. So this is the only species which live in the freshwater condition. Otherwise, rest of all of the species in the phylum Porifera are marine. Next character we'll see, that is habit. So basically this organism, where they live, the condition where actually they are living. See, this phylum Porifera, they do not have any type of locomotory organ. So they are always bottom dweller. That means they live in the seabed or ocean bed. So that type of organism which do not have any locomotory organ, and they attach with some type of substratum. Substratum it may be the seabed, it may be the sand, or it may be the rock also, or maybe suppose some another species too. So what is happening here? The habit, this organism generally are sessile animals. Sessile means, this word means, these organisms, they attach to the substratum. They live in the seabed or the riverbed. If it is the Spongilla, they live in the riverbed. Rest of the other organism, they live in the seabed or ocean bed. So this type of animal, they are sessile animal. Next characteristic, see, first of all, the symmetry. So under that symmetry, what we have seen, that this organism, they are not going to have any symmetry. So they are asymmetrical, but we have seen, I told you that in one class in the phylum Porifera, they have radial symmetry. Like you can see this organism. It's possible that you can divide this animal, you will get a circular cross section. Then after that you can have the radial symmetry. So the symmetry, they are asymmetrical, but yes, we have radial symmetry also. So where this radial symmetry you can see in the class Calcarea, where you can see Gransia. This is a species sponges. These are actually the radially symmetrical animal, Gransia. Next one, see their level of organization. So what is the level of organization? That means their body is made up of, see, the most simplest organism. So that's why they're going to have only the cellular level of organization. They're going to have just aggregation of cell. They don't have any type of tissue formation or organ formation. Next one, see their body form, body structure. See, so in that body formation you can see they are the species which actually have, suppose this is the species, okay. Now in that species they will have pores, okay, from outside. Inside they will have a cavity and they will have an opening too. So this is the body form. First of all, if you see this organism, these organisms are hollow. Hollow means this organism, they have a cavity inside. Okay, so cavity will be present in that animal. This organism is also porous. Porous means throughout their body they will have the pores. So with the help of the pores, they will take the gases and the nutrients in the inside and allow the waste product and the carbon dioxide to remove outside. So this is their body form. Next one, see their body layers. That means in this organism, will you see any germ layer? So you can see these are just the aggregations of cells. This organism are incapable of forming any germ layer. They do not have any germ layer, but yes, this organism will have some layers. The body will have some layers. So what are those layers? See, this organism will have the outermost layer. Suppose I will draw here itself. So this organism will have a layer here and inside another layer. So I will draw in detail the structure. See, this is the outer layer I am drawing. So in the outer layer you can see some flat cells are present. And what is that flat cells known as? These are known as pinacocyte. And there are some oval shaped cells are also present. You can see there are oval shaped, they are facing each other. So what are those flat cells known? These flat cells are known as pinacocyte. So these are the pinacocytes and some flat cells are present due to what actually pores are developed. They are known as the porocyte. So pinacocyte and the porocyte, they form the outer layer. What is that outer layer known as? This outer layer is known as pinacoderm. So this is the outermost layer of that phylum Porifera. The outermost layer is the pinacoderm layer. Inner layer. What is the inner layer? Inner layer is this layer. This layer I am drawing. This layer is made up of some other cells, and those cells actually they are ciliated cells. These cells are known as the schwannocytes. Okay, this layer, the inner layer, that is known as schwannoderm layer. So this organism, how many layers they have? Two layers. One is outer pinacoderm and inner schwannoderm. Pinacoderm is made up of two types of cell. One is porocyte. They are the oval shaped cell because of what they develop the pores. So throughout the body, obviously they will have porocyte because of what pores have been developed. And another layer, that the same outer layer, they will actually have the flat cells, and those flat cells are known as pinacocyte. Pinacocyte and porocyte will form the layer that is pinacoderm layer. Next layer, C, that is inner layer, or that innermost layer also we can call. The two layers only, outer pinacoderm and inner schwannoderm. So that schwannoderm is made up of one ciliated cell that is known as we can call these are the schwannocytes. So what is it? See, what is the reason why they have that ciliated cells also? See, this is the organism which is living in the seabed or the river bed. Now this organism, if they cannot move, how they will get the food? Obviously with water they have to enter. In the seabed the water current is also very less, or there is no water current. So wherever the water is there, that will remain there only. It will not coming to to them actually. So that's why what happened inside that organism, actually beating of cilia or beating of those schwannocytes occur. So whenever beating occurs, there is a current of water develop around them. So because of that, actually the water enters inside that organism. So water actually enter inside. Because of that, because of the reason is schwannoderm. Schwannoderm, which made up of the schwannocytes which beat, and due to the beating of the schwannocytes, a water current is developed, and due to that water current, actually they can trap the food and the food nutrients and the gases inside, and because of that actually they can live. They can perform digestion, respiration, circulation, excretion with the help of the same, that is schwannocyte. Now just see what is present in between this schwannoderm and the pinacoderm. See, in between they do not have any layer, that is actually one non-cellular jelly-like matrix, and that middle non-cellular jelly-like matrix, this is known as mesohyl, or we can call this is as the mesenchyme. So what is it? So this mesohyl layer, it's a non-cellular layer. It do not have any living cell of its own, but the cells from the pinacoderm or from the schwannoderm can live there. So this mesohyl layer basically made up of two components. One is the sponsin fiber. So here you can see whenever those organisms they die, what remains? The sponsin fibers. So because of what you can have those those spons, that is actually the sponsin fiber. So that sponsin fiber is present in the mesohyl layer. So sponsin fiber is present, and along with that they will have a very unique character of having some spicules or some spine-like structure. So those spicules or spines actually protect that organism from the predators. So predator directly cannot catch them because it is very spiny. What is the reason? Because this mesohyl layer is going to have the spicules or those spine-like structure which is present in the middle non-cellular layer. Now another important thing you can see here, that in this mesohyl layer some amoeba-like cells are present. So that amoeba-like cells, they are known as amoebocyte. So what is that amoebocyte? These are actually some living cells. These are not of this mesohyl layer's own. So these are coming, they're actually moving from here to there. They're actually helping in phagocytosis. They take the food and remove the waste product. So that actually for transportation, those amoebocytes are present. Now this amoebocytes, they will develop, they will modify into some different varieties of cells, and those cells will see all of the cells, their name, functions are important. So those amoebocytes are developed, modified into the following cells. So see, those amoebocyte I told, that is present in the middle non-cellular layer, that is mesohyl layer, that is developed into a number of varieties of cells. So what are those? The first cell I have showed here, I have written already. What are they? First one, this is trophocyte cell. Trophocyte, troph, autotroph, heterotroph. What are they? Troph means the nourishing part. So what is it? The trophocyte is the nourishing cell. Next one, thesocyte, this actually store the food granules. Next one, archaeocyte, this came a number of time in the exam. What is that archaeocyte? That is the totipotent cell. Totipotent cell means if suppose somehow some another type of cells they die and cannot perform the function, those archaeocyte will go to that region and perform that same function. So that is known as totipotency. So in case of archaeocyte, they have this totipotency so that it can transform into any of the type of cell. Next one, see gland cell, they produce the slimy substance over the organism. They produce a gelatinous substance. Next one, spongioblast. The spongin fibers, they will be actually produced by the spongioblast, and those spicules, they are formed from the scleroblast. So scleroblast, they will secrete the spicules. The spicules can be of 3 different types. What are they? First of all, this is monaxon type of spicule. Next one, triaxon type of spicule, and then the tetraxon type of spicule. So if it is going to have only one sting, then it is monaxon type of spicule. If it is going to have 3 stings, then it will be known as the triaxon type of spicule. And if it is going to have 4 spicules, four suppose stings, they are known as tetraxon type of spicule. So these are actually secreted from the scleroblast. Next one, collagen, they will secrete the collagen fiber. So collagen fiber is a very much important fiber that should be present in every animal, and in animal kingdom the most abundant type of protein is the collagen protein. So that is secreted by one cell, that is collagen. Next one, myocyte, which will produce the osculum. So remember, I will tell you what is osculum later on. Just from now, remember that will form the osculum. Next one, germ cell, it will produce the gamete. Both the sperm and the ovum, they are secreted by this gland cell. See, in case of phylum Porifera also, they are going to be the sexually reproducing animal. So in case of the sexually reproducing animal, they need to produce the gamete, the sperm and the ovum. So sperm and ovum, they are secreted actually from the germ cell. Next one, next cell is the chromocyte. So chromocyte is actually one type of cell which contains some pigments, due to what different colors can be seen. See, in case of phylum Porifera, you will see a variety of color, and those colors are because of this chromocyte which secrete the pigments. And the last side, remember, this is phagocyte. So it will help in phagocytosis. See, phagocytosis means what? First of all, suppose this is one type of particle. Now this phagocyte will come to that cell, suppose substance. It will engulf it, and after that it will take it to any of the location, and after that it will transfer. So this phagocyte help in suppose transportation of different nutrients. Suppose oocyte have trapped suppose some food, so that food will be transported to the another region by this phagocyte. Next one, suppose some dead cell, suppose some cells died, so those dead cell will be removed outside, removed by the body by those phagocyte. So these are the different cells that derived actually from the amoebocyte. So those are the cells we have discussed. Now after that we'll see a very unique characteristics of the phylum Porifera. Now just see a very unique characteristics of the phylum Porifera, that is canal system. So what is canal system? This is actually one water current system by which this organism trap food. This organism actually inspirate, that means take the gases, remove the gases. Then after that it will also help in expiration, that means excretion, that is removal of the waste product, as well as it helps in the transfer of the gametes, that is the gametes means basically sperm. Just see how many types of canal system can be seen. The most simplest type of canal system, this is known as Escon type of canal system. So let's just see what is actually the Escon type of canal system. I told already that it is this type of organism. This is actually porous organism, porous animals. That's why this organism is known as Porifera. Now just see, this organism is going to have numbers of pores. I told why those pores, how actually those pores are developed, due to the porocytes. Now just see, this is an organism. So those holes, these are the holes that have been present, you can see. So this is another hole that has been developed. So those pores, smaller spores throughout the body, they are known as the ostia. There is a large gastrovascular cavity developed that is known as sponzocyl, and the large opening here, this is known as osculum. So just see how this Escon type of canal system works. First of all, water along with nutrient, along with the food and the gases, that is oxygen, that will enter inside the body with the help of the ostia. Then after that it will go to the sponzocyl taking the nutrients. So the schwannocytes will grab this nutrient, and after that the cells will take the gas oxygen also. Then after that in the same water they will remove the waste product and carbon dioxide. So that carbon dioxide and the waste that will be present in the water in the sponzocyl. Now that waste and carbon dioxide with water that will be removed outside with the help of this osculum. So osculum remove the actually waste material and that gas carbon dioxide with the help of the osculum. So that is the most simplest type of canal system, that is Escon type of canal system. Now in the Escon type of canal system some type of problems appeared. Problems means this organism can trap that water for a very small period of time. So gradually what happened, to accumulate that water and to stay that water for a long period of time inside their body, now some invagination start to occur. That means now this organism start to develop this type of invaginations. Now this invaginations have, like suppose this is the organism, here also this has been developed. I have drawn only one part. See, so this organism first of all will have this opening, and this opening is known as ostia. So this is the ostia now. So this ostia will help in the trapping of that water with the nutrient and the gas. Then after that you can see there is a hole present. This is that hole. This hole is known as prosopyle. And there is another hole present here that is known as apopyle. Remember, this is the ostia, this is prosopyle, this is prosopyle, and this is the apopyle. So first of all, apopyle, yes, and there are two canals are also here. This is one canal, this is another canal. So this canal is known as incurrent canal, and this canal, this one which is from inside, that is known as the radial canal. Now just see how this water will enter inside the sponzocyte. First of all, with the help of the ostia, the water enters, it will reach the incurrent canal, here, this is the incurrent canal. From the prosopyle, it will go to the radial canal. From the radial canal, the water ultimately go to the sponzocyl via this apopyle. Through this apopyle, water will go to the sponzocyl. So due to that, actually it can check on that water, it can trap the water for a long period of time so that it can get enough time so that it can accumulate the gases and the nutrients. So in the sponzocyl they trap the water, then after that it takes the same function, it will take the gases, nutrients, remove the waste product and the carbon dioxide in the same sponzocyl. Now with the help of this osculum, they will remove the waste product. So this is the next type of canal system, that is known as second type, that is Sycon type of canal system. Now the last type of canal system, it is even more and more complex. That means invaginations now have occurred, developed from both the sides. That means from internal sides also as well as from the external side. So you can see, suppose these are some channels have been developed. So those channels, they are known as flagellated chambers. So these are the flagellated chambers. So from outside also this organism will take the gas, nutrients, and this way the whole organism will develop this type of flagellated chambers. So what happened? First of all, the water will enter inside the body with the help of the same, that is ostia. From the ostia, it will go to the same incurrent canal. But from the incurrent canal now through this prosopyle, it will go to this flagellated chambers. These are the flagellated chambers. Now from the flagellated chambers, it will go to the next part, that is from the apopyle. So these are the apopyle. Then after that, from the apopyle, it will not go to the spongiosum. It will be now known as the excurrent canal. So from the excurrent canal, it will go outside. So the whole diagram will be a little complex also. I have given in the PDF, so you can check the PDF so that you can understand in a better way. So these are the channels actually. They are present like that. From outside also invaginations have occurred. So this way the whole organism would develop. So that how actually they will take the gas and nutrients with the water is, first of all, with the ostia, it will take the water, it will go to the incurrent canal. From the incurrent canal, it will go to the prosopyle. From the prosopyle, it will go to this flagellated chamber. So these round structures, they are the flagellated chambers. From the flagellated chambers, water will ultimately come outside through this opening. So this opening is known as the apopyle. So here those are the apopyles. From the apopyle, it will go, the water will go to the excurrent canal. Now from the excurrent canal, the water will be removed outside. So this last type of canal system, that is known as the Leucon type of canal system. So these are the three different type of canal system we have seen. Now just see the functions of canal system. First function of the canal system is to get the food. Actually for food gathering, the canal system works. Next one, for the exchange of gases, that means it will take the oxygen inside and remove carbon dioxide with the help of the same canal system. The third one, that is removal of the waste product. So removal of the nitrogenous waste product will be by the same, that is the canal system. And the fourth very important function, that is for the transfer of the sperms. So the sperms that is produced by one suppose organism, that will be trapped, that will come outside with the help of the osculum, and it will go to the water. Then a different organism with that water, if the sperm enters, the spermatocytes will take that sperm. So from a different organism, the sperm will be trapped, and in the mesohyl layer, the sperm and the ovum will fuse. Okay, so this way the canal system will work. So after that, the next type, after the general characteristics we go on. So the next type that is important also, that is the digestion. See what type of digestion they will have. Digestion will be simply intracellular. So in case of this organism, they do not develop any type of digestive system or canal or anything like that. They will just have the cell. Inside the cell, they will get the food, digest inside the cell itself. That is intracellular type of digestion. Now their skeleton. See, in case of them, do they have any type of skeleton? So actually no, this organism do not have any type of bony skeleton. Instead, what they have spongin fibers and spicules. Remember, I told in the mesohyl layer that in case of the middle non-cellular mesohyl layer, they will have the spicules. So their spicules, they will actually help this forming this skeleton, that is actually endoskeleton. Then remaining functions, that is respiration, then excretion, all these functions, they are performed by the same canal system. Now next just come to the reproduction. In that reproduction, what you will see, that this organism will prefer sexual reproduction, but then also the asexual reproduction can also be seen, like fragmentation budding. Like suppose this is the organism, they will develop a bud, then this bud will grow in size, then after that they get separated. So asexual reproduction can also be seen, but yes, sexual reproduction is the most favorable one. Then after that, what type of fertilization they will have? does that fertilization internal or external? See, fertilization is internal. That means inside the body, the meeting of male and female gamete occur. So fertilization is internal. What about development? Development is indirect. Indirect means in case of them, the larvae are present. There are two larvae present. One is known as parenchyma and another is the amphiblastula. So these larval stages are present. So those larvae will help actually the larval stages which actually get develop, metamorphose into the adult. So this is all about the phylum Porifera. Actually, in this phylum, we can see all these things, and from the canal system question are been asked lot of times in NEET also. And remember those cells I have told, the amoebocyte are developing, the trophocyte, testocyte, gland cells, scleroblasts, all those cells and their functions, they have been very much important. And the last point which is very much important is their common name and scientific name. Like I can give you some examples, like Eusponsia. This is one species which is useful as a bath sponge. That means used as a bath like a scrubber, we use the Eusponsia. Why you are using is the reason is they actually lack the spicules. So as they don't have the spicules, they are not going to sting us. So that's why it is used in bath. So this is commonly known as bath sponge. Similarly, we have Euplectella, which is known as Venus flower basket. Next one that is Hyalonema, which is also known as glass robe sponge. Then we have the Chelina, Cliona. Cliona is also lots of common name and scientific names are here. You just check the books. In the book, in NCERT book, you can get all those common name and scientific name. And in NEET, if question come from common name and scientific name, it will come only from NEET. Otherwise, in different other reference book, you can get a variety of species. You can not remember all the scientific and common names. So better you can stick to NCERT. In NCERT, you will get common name and scientific name. Only from that point only you can go for that NCERT. So this is all about the phylum Porifera. So we have completed the general characteristics. They're all characteristics, canal system I have told, reproduction. One more thing, their general and scientific name and their general name, all of you see, this is very much important. So this is all about phylum Porifera. Thank you.